Welcome to my channel Deep Learning Biology. My name is Dr. Vikas Mahardwaj. Now in this video I am going to teach you about amino acids. So here in this video you are going to learn basic things related to amino acids. Now before going further, I just want to ask you one simple question. Very simple. Just try to think about a cell. What do you think? What are total number of amino acids present within a cell? What are total number of amino acids? Okay, you know there are different types of amino acids, and what do you think? What are total number of amino acids present within the cell? What do you think? No. Keep in mind, within the cell you are going to see more than 300 amino acids, you can say types. Try to see here. You know very well, within the proteins you are going to see amino acids. Proteins, these are most abundant biological macromolecules, and you know very well that proteins generally they are polymer of amino acids. In proteins, one amino acid is joined to another amino acid through peptide bond. So here, if you see in proteins, generally you are going to see the presence of amino acids. Those amino acids are proteinogenic amino acids. The amino acids which are present within the proteins, such amino acids, they are known as proteinogenic amino acids. But within the cell cytoplasm, you are also going to have other amino acids which do not take part in formation of proteins. Such amino acids are known as non-proteinogenic amino acids. So here keep in mind, one is proteinogenic amino acid, another is non-proteinogenic amino acids. So proteinogenic amino acids, they are present within the proteins. Non-proteinogenic amino acids, they are going to have different roles. Okay, so here keep in mind, total number of amino acids which are going to be present within the cell, they are more than 300 amino acids and approximately 22. Okay, here try to see, earlier we used to have 20, now we are going to have 22. So 22 amino acids, they are proteinogenic amino acids and they are present in the proteins. So hopefully all the points are clear to you. Now let us move forward. So here hopefully you know that amino acids, what they are going to have? Carboxylic group, COOH group. They are going to have amino group, NH3 plus group. In amino acid you are going to have R group and you are going to have hydrogen atom. Just have a look into the structure of any amino acid. Okay, try to see what you are going to see. One carboxylic group, COOH group. Keep in mind, this COOH group is going to have carbon and this is the first carbon. Okay, the first carbon is attached to another carbon and that carbon is alpha carbon. Okay, now this C alpha, it is attached to one hydrogen atom and one R group. Okay, yes, in case of glycine you are going to have two hydrogen atom and here the fourth one is the amino group. So here you see in the amino acid what you are going to have: carboxylic group, amino group, R group, hydrogen atom. Now here what you are seeing, that the C alpha, it is being attached to four different atoms, and here you see the C alpha. Okay, on one side you are going to have carboxylic group, on another side you are going to have amino group. So here on one side you are going to have acid COH, another side you are going to have amino group. That's why what we are going to say it amino acid. Keep in mind here the C alpha, okay, the alpha, okay, the carbon, it is attached to carboxyl group and amino group. So here all the amino acids, they are going to have this parent structure, and all these amino acids, as they are having the alpha group attached to COH group and amino group, that's why these amino acids, they are known as alpha amino acids. Okay, so here generally what we write, that alpha amino acids. So here keep in mind the alpha group, the C alpha, it is going to have COH and the amino group. Okay, so hopefully now it is clear. Amino acids, generally what they are going to have, carboxylic group, amino group, and here the C alpha carbon, okay, it is going to attach with COH and the amino group. Yes, here also we are going to have hydrogen and the R group. Now here keep in mind, based on different type of R group, okay, so you are going to have different amino acids. So we are going to discuss about different types of proteinogenic amino acids, and what we are going to see, you will see that all these amino acids, they are going to have different R group. Now let's move further. So here if you draw the structure of this alpha amino acid, so here you are going to have COH group, on the another side you are going to have amino group, okay, R group and the hydrogen. Now here you see the C alpha, it is going to act like a chiral center. It is going to act like a chiral center. So here you see the alpha carbon, it is being attached to 4 different atoms. Okay, so it is going to act like a chiral center. It is asymmetric, and if you are going to draw the stereoisomers, hopefully you know what are isomers. Okay, if you are going to draw stereoisomers, means you are going to have different shape in the space. Okay, so here when you are going to have stereoisomers, in this case the C alpha, okay, as it is chiral, it can show you 2 possible stereoisomers, and all of these 2 possible stereoisomers, they will be non-superimposable mirror image. Now try to see here. Here let us draw the structure. Okay, so here C alpha, it is attached to, on upper side you are going to have COH, lower side amino, on one side we have shown the hydrogen, another side you have shown the R group. Just try to draw the mirror structure. Okay, now here you see C alpha, COH, NH3 plus, HR. Now both, if you see, okay, here we have put one mirror. Okay, now here if you see, what you are going to see, that both of these images, they are non-superimposable, just like your hands. Okay, you can't put this right hand, okay, this is left, okay, this, so these are non-superimposable mirror image. So such type of isomers, stereoisomers, they are known as enantiomers. They are known as enantiomers. Okay, non-superimposable stereoisomers, they are known as enantiomers. Now here if you see the C alpha, it is asymmetric. Okay, C alpha, as it has been attached to 4 different, okay, so it is asymmetric, it is chiral, and when you are going to have such situation, in this situation the compound, okay, it is going to be optically active. Okay, so here you see that when you are going to have C alpha, if it is asymmetric, if it is chiral, okay, now these chemicals, you can say, they can rotate plane polarized light. Okay, so if you are going to prepare a solution of such compound, okay, then what you are going to see, if you are going to allow the plane polarized light to pass through, okay, then the light either may go in right direction or it may go in the left direction. So now we use the symbol plus or minus. Okay, so the plane polarized light, either it can move in the right direction or it can move in the left direction. Okay, so keep in mind that these amino acids which are going to have asymmetric carbon, what they are going to be optically active, and they can rotate the plane polarized light either in the plus direction or in the minus direction. Now here, if you see the case of glycine, try to see in the case of glycine, the C alpha, okay, it is attached to two hydrogen atom. So you are going to have carboxyl group, you are going to have amino group, and here you are going to have two hydrogen atom. You can say R group here is absent. Okay, so here the R group is H hydrogen. So here you are going to have two hydrogen. Here the C alpha, it is not attached to four different atoms. So two are same. So in this situation, keep in mind, it is not going to be a chiral center. It is not asymmetric. And when it is not chiral asymmetric, then keep in mind it is not optically active. Means if you are going to have a solution of glycine, okay, and if you are going to pass the plane polarized light, the light, it is not going to move in either positive or negative direction. Okay, so keep in mind glycine is not optically active. Okay, now let us move forward. Let's take the case of alanine. Okay, alanine, try to see here, it is going to have amino group, it is going to have this C alpha, and then you are having hydrogen, then you are going to have carboxyl group. At the bottom you are going to see the R group. Now try to see here. Okay, this is the chemical structure of alanine. Now here what I have shown you, that amino group, okay, try to see it is present on the left side. Okay, so when the amino group, it is present on the left side, so you can say this is L-alanine. This is L-alanine. Now if you draw the structure like this and you are going to put the amino group on the right side, okay, now this is going to represent the D-alanine. Okay, so here one is L-alanine, another is D-alanine. Now try to see here. Here keep in mind D and L, they are going to represent absolute configuration. Keep in mind DLL, they represent the absolute configuration. Okay, now here I also want to tell you, if you are going to have D amino acid, okay, so it can rotate the plane polarized light either in plus or in the minus direction. Same way, if you are going to have L amino acid, it can either rotate the plane polarized light in plus direction or negative, minus. Okay, so earlier we used to have the symbol like dextrorotatory, levorotatory, okay, small d, small l, okay, but try to see here. Okay, now we are going to have this, okay, plus and minus. So try to see here the D amino acids, okay, keep in mind they can rotate the light either in positive or in negative direction. Same way L amino acids, they can rotate the plane polarized light either in positive or negative direction. Okay, so here keep in mind small d, small l, okay, it represent, earlier we used to have this symbol small d, okay, for positive, small l for negative. Okay, now we use plus and minus. Okay, so rotation of light, okay, that is another. Okay, so you can't say if there is an amino acid which is going to have configuration, okay, D, okay, it will rotate the light in positive. It can rotate the light in both positive or negative. Same way, the amino acid which is going to have the configuration L, okay, they can rotate the light either in positive or negative. Okay, just make it clear. Now here if you see, generally in proteins, what you are going to see, you are going to see L amino acids. Okay, generally if you see naturally in proteins, the configuration of amino acid is L. And here keep in mind, L doesn't mean these are laboratory. Keep in mind, not all L amino acids are laboratory. Okay, say there can be positive or negative. They can, L amino acids, they can rotate the light either in positive direction or in negative direction. So hopefully this point is clear to you. Now let us see here. Try to see, we are going to discuss the classification of amino acids based on R group. Okay, so here you see, if you remember, in amino acids we are going to have different R group. Based on R group, okay, based on R group, you are going to have different groups of amino acids. One is you can have non-polar aliphatic. Okay, in some of the amino acids, what you are going to have aliphatic chain. Okay, and these amino acids are actually non-polar hydrophobic amino acids which are going to have aliphatic R group. Yes, you are going to have aromatic R group. Here also you are going to see hydrophobic amino acids. Okay, you can say these are also non-polar. Yes, the R group, based on R group, you can have the polar. Okay, and you know polar, they are hydrophilic, and here the polar, they can be further classified in such a way that in one condition the functional group, they are uncharged. Okay, yes, there can be positively charged or there can be negatively charged. So based on R group, you are going to see these classification. We are going to discuss each one by one. Now try to see here, non-polar aliphatic R group. So the amino acids which are non-polar, and they are going to have aliphatic R group, and generally keep in mind these are hydrophobic, means they don't love the water, they want to remain away from the water. Now here if you see the examples, okay, glycine, okay, glycine, alanine, proline, valine, leucine, isoleucine, methionine. Glycine, alanine, proline, valine, leucine, isoleucine, methionine. These are examples of non-polar aliphatic, you can say hydrophobic amino acid. Now try to see here. Here you see alanine, here you see the valine, and here if you see leucine, and if you see this one isoleucine. Okay, now all these amino acids, okay, yes, they are going to have non-polar aliphatic R group. Try to see here, if let's suppose there is a protein and if it is present within the cell cytoplasm, if you try to think about the outside environment, it is hydrophilic. Okay, so you know within the cell cytoplasm we are going to have 70% water. So here in this situation, when we are going to have hydrophilic environment, and these are hydrophobic amino acids, the hydrophobic, they will tend to move away from hydrophilic things. Okay, so they tend to move away from water. So here keep in mind these amino acids, they will tend to cluster together and they will be inside. Okay, and this is going to happen due to hydrophobic effect. Okay, so these amino acids, alanine, valine, leucine, okay, isoleucine, they will try to cluster together and they will be present inside the protein. In case of cytosolic proteins, then it is being found. Now glycine, you know very well, it is going to have sweet taste. Now we are going to have some more discussion related to proline and methionine. Try to see here, in the case of proline, keep in mind, in case of proline, you are going to have distinctive cyclic structure. Just you can have this inverted P, okay, like structure. And if you want to see, you can draw the structure like this. Okay, so if you are going to draw the structure of proline, what you are going to see in proline, you are going to see it is actually the presence of amino group. Here you are not going to have amino. Here what you are going to see, amino. Actually proline is a amino acid. Okay, so here one thing what you are going to see, presence of amino group, and it is going to have distinctive cyclic structure. Okay, so here the rotation of bonds, it is not free. So here keep in mind, proline, okay, it is going to have rigid conformation. Okay, so wherever within the protein the amino acid proline is present, okay, so wherever this amino acid present, okay, the protein, it is not going to have at that point structural flexibility. So keep in mind, proline, wherever it is present within the protein, okay, it will lose, okay, it will not allow the protein to move freely. Okay, rest of the amino acids, they can move freely. Okay, so here keep in mind, proline, it provides rigid conformation, it reduces structural flexibility of the protein. Now let us move further. If you see the case of methionine, okay, now try to see here, methionine is sulfur containing amino acids. Keep in mind, if you see these proteinogenic amino acids, there are two amino acids which are going to have sulfur. One is methionine and the another one is cysteine. Now here let us discuss the first one, the sulfur containing amino acid. Okay, here if you see in the case of methionine, here what you are going to see, presence of sulfur. So here you see, you can divide it into two portions. Okay, both they are being joined together by the sulfur. So here in between you are going to have this sulfur. Now here you see, this is arrangement like this, and this is going to represent the thioether group. In methionine, what you are going to have, thioether group. And here keep in mind, thioether group, it is going to form stable bond. It is a stable bond. Okay, so that is very important. Try to see here, methionine is a sulfur containing amino acid, okay, and it is going to have a stable thioether group. That's why here you will see that methionine is not going to form part in formation of disulfide linkage. Okay, so here this point is very important, that methionine, yes, it has sulfur, okay, but this sulfur is not going to take part in formation of disulfide linkage. Which one is going to form? The cysteine, the another amino acid, sulfur containing amino acid, okay, is cysteine, that take part in formation of disulfide linkage. Okay, so hopefully this point is clear to you. Now here if you see the another group, what you are going to have, aromatic R group. Here in this class what you are going to have, phenylalanine, tyrosine, tryptophan. Phenylalanine, tyrosine and tryptophan. Now try to see here, in case of tyrosine, if you just saw the structure, what you are going to see, the OH group. Okay, so OH group is present there, and this OH group, it takes part in formation of hydrogen bonds. So in many enzymes, the tyrosine is involved in formation of hydrogen bonds. This tyrosine amino acids is involved, okay, in carrying out various reaction. So so keep in mind tyrosine is going to have this OH group, okay. Yes, if you see these amino acids, phenylalanine, tyrosine, tryptophan, and if you see the structure, they are going to have aromatic group, okay, benzene ring like structures, and these structures, they can absorb light, okay. And here if you see, we can quantitate proteins at 280 nanometers, okay. Generally if we have to see whether in a sample proteins are present or not, generally we use UV spectrophotometer, and what wavelength of light we are going to use? 280 nanometer. For nucleic acid we are going to use 260 nanometer. Now here keep in mind, due to the presence of tyrosine and tryptophan, majorly tyrosine and tryptophan, okay, the proteins, they are going to absorb light, and generally this absorption of light happens at 280 nanometers, and this helps in quantitative measurements of proteins, okay. Now let us move further. Now here we are now going to discuss polar uncharged amino acids. So I have told you, okay, one can be uncharged, another can be positively charged, another can be negatively charged, okay. Now these polar ones, amino acids, generally they can form hydrogen bonds with water, okay. So here you are going to see examples like serine, threonine, cysteine, asparagine, glutamine. Serine, threonine, cysteine, asparagine, and glutamine. Now try to see here. In serine and threonine, what you are going to see? Polar OH group. In case of cysteine, keep in mind it is a weak acid, and here it can form hydrogen bonds with oxygen and nitrogen. And if you see the case of asparagine and glutamine, here you are going to have polar amide group. So here in these amino acids, what you are going to have? Polar groups which can form hydrogen bonds with water, and here these functional groups are uncharged, they are not going to have charge. Now here if you see this amino acid, okay, now this is cysteine, okay. Now if you see the case of cysteine can, you can say, join with another cysteine, okay. And here if you remember, I have told you in cysteine what you are going to have? The sulfur, okay. So here you see, thus when cysteine is going to join with another cysteine, there will be formation of disulfide linkage. So here keep in mind, there are two sulfur containing amino acid, one is methionine, another is cysteine, okay. Two cysteine, they can join together, and they are going to, there is in between formation of disulfide linkage, okay. So it will lead to formation of cysteine, okay. Now here what we are going to see? Formation of disulfide linkage. If in any protein you are going to have disulfide linkage, that is going to give stability to the protein, okay. So here you see in between cysteine, okay, two cysteine, they can join together via disulfide linkage, and what will be the formation of cysteine, okay. So hopefully this is clear to you. Now let us discuss the positively charged R group containing amino acids. So here you are going to have amino acids which are going to have positively charged, and these are polar, and generally these are actually the basic one. Generally at pH 7 they are going to have positive charge. So here the examples comes lysine, arginine, and histidine. Lysine, arginine, and histidine. Now try to see here. In the case of arginine, what you are going to see? Positive guanidino group. In arginine, what you are going to have? Positive guanidino group. In case of histidine, you are going to have positive imidazole group. Okay, so in histidine you are going to have positive imidazole group. Now here I also want to tell you more about this histidine. Keep in mind, in case of histidine, the pKa pKa value is near, okay, to neutral pH. Okay, its pKa value is very similar to neutral pH value. At the neutral pH, it can either lose or accept the protons. Okay, so here you try to think, the histidine is going to have neutral pKa value. So at a neutral pH, at the cellular pH, it can easily donate proton, it can accept protons. Okay, so this is going to facilitate many of the enzymatic reactions. So in many of the enzymes active site, what you are going to see, the presence of histidine, and this histidine either can accept and donate protons. Why? Answer is simple, because in the pKa value for the histidine, okay, it lies at neutral pH. So at neutral pH, histidine can easily accept and donate protons. That's why in many of the enzymes where you are going to have presence of histidine, okay, they can actively act at neutral pH. So this histidine facilitate many of the enzyme reaction at neutral pH. Hopefully this point is clear to you. Now at the last, okay, we are going to discuss negatively charged amino acids. Negatively charged amino acids which are going to have negatively charged R group. Okay, at pH 7, generally these are going to have negatively charged R group, and generally these are acidic amino acids. For example, aspartic acid and glutamic acids. So hopefully through this video you have learned clearly the basic points related to amino acids. Thank you for watching this. Please do like, share the video and subscribe to my channel Deep Learning Biology. Bye bye. Take care.